and the cells are distributed depending on the gene expression. So you can see here, like the cells that are coming from the primary tumors, they are different to the PDO cells, and yeah, so this is why they are distributed differently. So we wanted to characterize these cells a bit better to see why this is happening. So we look for epithelial markers, and first here we have ECAN. So ECAN is widely expressed in primary cells and also in the organoids. And then we look at the keratin 19, that is a luminal marker, and we find out that this is expressed in the primary cells and also in most of the PDO cells. And then we look at the basal marker, that is keratin 14, and we find out that this is expressed in the PDOs, but it's not expressed, or at least not widely expressed in the primary cells. And then we look at the estrogen receptor, and we found out that this is lost in the PDOs. So we wanted to know like why or how different these tumor primary cells and these PDO cells are. And we look at some biological processes. So we look at this and we saw that the similarities are driven by the type of meat and not by the origin of the primary tumor. So if we look further only at the organoids, we could see that the cells that are in type 1, they express genes, or they have like higher expression of genes that are related to EMT and invasion, and the cells that are in type 2, they have a higher expression of genes that are in hypoxia and inflammation. So then if we look at the cell distribution, we can do like different clusters, and this is just depending on the gene expression, independently of the sample or the culture. And then if we compare these clusters with the origin of the culture, we can see like some of the clusters, they have cells that are coming from the primary cells, but also from the organoids. So this means that some of the cells that are in the organoids express some gene expression patterns that are similar to the primary. It's just that they are so few that you cannot see them like at the first look. Then if we look at these clusters and we compare them with the samples, we can see that also like most of the PDO samples, they have some cells that are also like expressing like these gene expression patterns that are similar to the primary, especially this number 22. Sorry, here is wrong. So it's the number 22A, and this has a bit more fewer cells, a bit more cells than the other samples that are like similar to the primary. So if we go back to the loss of estrogen receptor, like I said before, this estrogen receptor is only expressed in luminal cells, so they also, and they are not expressed in the basal cells, the ones that are expressed in this keratin 14. And if we look at the clusters that are formed before in this here map, we can see that there are some clusters where there are primary cells and organoid cells, and the estrogen receptor is expressed. But this means that not all the cells in the cluster are expressing estrogen receptor. They have similar gene expression patterns, and some of the cells there are expressing estrogen receptor. So then if we look further, if we look at the expression of estrogen receptor in all the cells, for example, in this cluster 23, is one that has more cells that are coming from the primary, that are coming from the organoids and also from the primary.